Greetings, fellow adventurers, and welcome back to my channel. Welcome, friends, to our video dedicated to Homeobox. Let's dive right in and uncover its wonders. A Homeobox is a DNA sequence, around 180 base pairs long, that regulates large-scale anatomical features in the early stages of embryonic development. Mutations in a Homeobox may change large-scale anatomical features of the full-grown organism. Homeoboxes are found within genes that are involved in the regulation of patterns of anatomical development morphogenesis in animals, fungi, plants, and numerous single-cell eukaryotes. Homeobox genes encode Homeodomain protein products that are transcription factors sharing a characteristic protein fold structure that binds DNA to regulate expression of target genes. Homeodomain proteins regulate gene expression and cell differentiation during early embryonic development, thus mutations in Homeobox genes can cause developmental disorders. Homeosis is a term coined by William Bateson to describe the outright replacement of a discrete body part with another body part, e.g. antennapedia replacement of the antenna on the head of a fruit fly with legs. The homo prefix in the words Homeobox and Homeodomain stems from this mutational phenotype, which is observed when some of these genes are mutated in animals. A Homeobox domain was first identified in a number of Drosophila homeotic and segmentation proteins, but is now known to be well conserved in many other animals, including vertebrates. As we venture forward, let's take a closer look at discovery and its impact on our understanding. The existence of Homeobox genes was first discovered in Drosophila by isolating the gene responsible for a homotic transformation where legs grow from the head instead of the expected antennae. Walter Gehring identified a gene called antennapedia that caused this homotic phenotype. Analysis of antennapedia revealed that this gene contained a 180 base pair sequence that encoded a DNA binding domain, which William Bateson termed the Homeobox. The existence of additional Drosophila genes containing the antennapedia Homeobox sequence was independently reported by Ernst Halfan, Michael Levine, William Bateson, and Walter Jacob Gehring of the University of Basel in Switzerland and Matthew P. Scott and Amy Weiner of Indiana University in Bloomington in 1984. Isolation of homologous genes by Edward de Robertis and William Bateson revealed that numerous genes from a variety of species contained the Homeobox. Subsequent phylogenetic studies detailing the evolutionary relationship between Homeobox-containing genes showed that these genes are present in all bilaterian animals. With that being said, let's now move on to Homeodomain structure. The characteristic Homeodomain protein fold consists of a 60 amino acid long domain composed of 3 alpha helices. The following shows the consensus Homeodomain amino acid chain. Helix 1 helix 2 helix RRKRTLYTRYKFNOYLTRHFNLTRHIKIWFQNRMKWKKN 10 20 30 40 50 60. Helix 2 and helix 3 form a so-called helix-turn helix HTH structure, where the 2 alpha helices are connected by a short loop region. The N-terminal 2 helices of the Homeodomain are antiparallel and the longer C-terminal helix is roughly perpendicular to the axis of the first 2. It is this third helix that interacts directly with DNA via a number of hydrogen bonds and hydrophobic interactions, as well as indirect interactions via water molecules, which occur between specific side chains and the exposed bases within the major groove of the DNA. Homeodomain proteins are found in eukaryotes. Through the HTH motif, they share limited sequence similarity and structural similarity to prokaryotic transcription factors such as lambda-phage proteins that alter the expression of genes in prokaryotes. The HTH motif shows some sequence similarity but a similar structure in a wide range of DNA-binding proteins, e.g., CRO and repressor proteins, Homeodomain proteins, etc. One of the principal differences between HTH motifs in these different proteins arises from the stereochemical requirement for glycine in the turn, which is needed to avoid steric interference of the beta carbon with the main chain, For CRO and repressor proteins, the glycine appears to be mandatory, whereas for many of the homotic and other DNA-binding proteins the requirement is relaxed. Get ready for an enlightening exploration as we dig into sequence specificity and understand its role in the broader context. Homodomains can bind both specifically and inspecifically to DNA, with the C-terminal recognition helix aligning in the NS major groove and the unstructured peptide tail at the N-terminus aligning in the minor groove. The recognition helix and the interhelix loops are rich in arginine and lysine residues, which form hydrogen bonds to the DNA backbone. Conserved hydrophobic residues in the center of the recognition helix aid in stabilizing the helix packing. Homodomain proteins show a preference for the DNA sequence 5TAAT3. Sequence-independent binding occurs with significantly lower affinity. The specificity of a single homodomain protein is usually not enough to recognize specific target gene promoters, making cofactor binding an important mechanism for controlling binding sequence specificity and target gene expression. To achieve higher target specificity, homodomain proteins form complexes with other transcription factors to recognize the promoter region of a specific target gene. As we venture forward, let's take a closer look at biological function and its impact on our understanding. Homodomain proteins function as transcription factors due to the DNA binding properties of the conserved HTH motif. Homodomain proteins are considered to be master control genes, meaning that a single protein can regulate expression of many target genes. Homodomain proteins direct the formation of the body axes and body structures during early embryonic development. Many homodomain proteins induce cellular differentiation by initiating the cascades of coagulated genes required to produce individual tissues and organs. Other proteins in the family, such as NA&OG, are involved in maintaining pluripotency and preventing cell differentiation. As we venture forward, let's examine regulation in detail and gain a deeper appreciation for its significance. Hox genes and their associated microRNAs are highly conserved developmental master regulators with tight tissue-specific spatiotemporal control. These genes are known to be dysregulated in several cancers and are often controlled by DNA methylation. The regulation of Hox genes is highly complex and involves reciprocal interactions, mostly inhibitory. Drosophila is known to use the polycomb and trithorax complexes to maintain the expression of Hox genes after the down-regulation of the pair rule and gap genes that occurs during larval development. Polycomb group proteins can silence the Hox genes by modulation of chromatin structure. Get ready for an enlightening exploration as we dig into mutations and understand its role in the broader context. Mutations to Homeobox genes can produce easily visible phenotypic changes in body segment identity, such as the antennapedia and bithorax mutant phenotypes in Drosophila. Duplication of Homeobox genes can produce new body segments, and such duplications are likely to have been important in the evolution of segmented animals. Let's now turn our gaze towards evolution and explore the fascinating connections it has to offer. Phylogenetic analysis of Homeobox gene sequences and homodomain protein structures suggests that the last common ancestor of plants, fungi, and animals had at least 2 Homeobox genes. Molecular evidence shows that some limited number of Hox genes have existed in the Anidaria since before the earliest true bilateral, making these genes pre-Paleozoic. It is accepted that the 3 major animal ANTP class clusters, Hox, Par-Hox, and NK meta-Hox, are the result of segmental duplications. A first duplication created meta-Hox and proto-Hox, the latter of which later duplicated into Hox and Par-Hox. The clusters themselves were created by tandem duplications of a single ANTP class Homeobox gene. Gene duplication followed by no-functionalization is responsible for the many Homeobox genes found in eukaryotes. Comparison of Homeobox genes and gene clusters has been used to understand the evolution of genome structure and body morphology throughout metazoans. Prepare yourself for an eye-opening discussion on Hox genes in the upcoming portion of this video. Hox genes are the most commonly known subset of Homeobox genes. They are essential metazoan genes that determine the identity of embryonic regions along the anterior-posterior axis. The first vertebrate Hox gene was isolated in Xenopus by Edward de Robertis and colleagues in 1984. The main interest in this set of genes stems from their unique behavior and arrangement in the genome. Hox genes are typically found in an organized cluster. The linear order of Hox genes within a cluster is directly correlated to the order in which they are expressed in both time and space during development. This phenomenon is called collinearity. Mutations in these homotic genes cause displacement of body segments during embryonic development. This is called ectopia. For example, when one gene is lost, the segment develops into a more anterior one, while a mutation that leads to a gain of function causes a segment to develop into a more posterior one. Famous examples are antennapedia and bithorax in Drosophila, which can cause the development of legs instead of antennae and the development of a duplicated thorax, respectively. In vertebrates, the four parleg clusters are partially redundant in function, but have also acquired several derived functions. For example, Hoxa and Hoxt specify segment identity along the limb axis. Specific members of the Hox family have been implicated in vascular remodeling, angiogenesis, and disease by orchestrating changes in matrix degradation, integrins, and components of the ECM. Hoxa is implicated in atherosclerosis. Hoxt and Hox are proinvasive, angiogenic genes that upregulate and are integrins and embryonics, respectively. Hoxa induces endothelial cell EC migration by upregulating and upper. Conversely, Hoxt and Hoxa have the opposite effect of suppressing EC migration and angiogenesis, and stabilizing adherens junctions by upregulating to downregulating upper and by upregulating downregulating VEGFA, EFNA, HIF-alpha and COX2, respectively. Hoxa also upregulates the tumor suppressor and act by downregulation of PTEN. Suppression of Hoxa has been shown to attenuate hemangioma growth. Hoxa has far-reaching effects on gene expression, causing genes to become upregulated upon its induction in breast cancer cell lines. Hoxa protein transduction domain overexpression prevents inflammation shown by inhibition of alpha-inducible monocyte binding to Homeobox. Brace yourselves for the next chapter, where we'll be dissecting LIM genes. LIM genes named after the initial letters of the names of three proteins where the characteristic domain was first identified in code to 60 amino acid cysteine and histidine-rich LIM domains and Homeodomain. The LIM domains function in protein-protein interactions and combine zinc molecules. LIM domain proteins are found in both the cytosol and the nucleus. They function in cytoskeletal remodeling, at focal adhesion sites, as scaffolds for protein complexes, and as transcription factors. Now, let's shift our attention to PAX genes. Most PAX genes contain a homeobox and a paired domain that also binds DNA to increase binding specificity, though some PAX genes have lost all or part of the homeobox sequence. PAX genes function in embryo segmentation, nervous system development, generation of the frontal eye fields, skeletal development, and formation of face structures. PAX6 is a master regulator of eye development, such that the gene is necessary for development of the optic vesicle and subsequent eye structures. Get ready for an exciting exploration as we unravel the mysteries of POU genes. Proteins containing a POU region consist of a homodomain and a separate, structurally homologous POU domain that contains two helix-turn helix motifs and also binds DNA. The two domains are linked by a flexible loop that is long enough to stretch around the DNA helix, allowing the two domains to bind on opposite sides of the target DNA, collectively covering an 8-base segment with consensus sequence 5-ATGCAA83. The individual domains of POU proteins bind DNA only weakly, but have strong sequence-specific affinity when linked. The POU domain itself has significant structural similarity with repressors expressed in bacteriophage, particularly lambda-phage. Get ready to immerse yourself in the world of plant Homeobox genes as we examine its impact and relevance. As in animals, the plant Homeobox genes code for the typical 60 amino acid long DNA binding homodomain, or in case of the TALE3 amino acid loop extension Homeobox genes for an atypical homodomain consisting of 63 amino acids. According to their conserved intranexon structure and to unique codomain architectures, they have been grouped into 14 distinct classes: HDZIPI-4, BL, KNOX, PLINC, WOX, PHD, DDT, NDX, LDS, AWADE, and PINTOX. Conservation of codomains suggests a common eukaryotic ancestry for TALE and non-TALE homodomain proteins. The spotlight now falls on human Homeobox genes as we delve deeper into its details. The HOX genes in humans are organized in 4 chromosomal clusters. Name chromosome gene HOXA, or sometimes HOX chromosome 7 HOXA HOXB chromosome 17 HOXC chromosome 12 HOXD chromosome 2 HOX genes are analogously found in 4 areas. They include and other genes considered HOX like include MOOX, and NK. The NK-like NKL genes, some of which are considered meta-HOX, are grouped with HOX-like genes into a large ANTP-like group. Humans have a distal-less Homeobox family, DUX, and DUX. DUX genes are involved in the development of the nervous system and of limbs. They are considered a subset of the NK-like genes. Human TALE3 amino acid loop extension Homeobox genes for an atypical homodomain consist of 63 rather than 60 amino acids: X, MKX, KNOX, TGIFLY. In addition, humans have the following Homeobox genes and proteins: Salim class, ISL, Lux, POU class, HDX, PALF, and PALF CRS class, HNF class, box for SINE class, 6 CUT class, Anikit Cux, SAP, ZF class, ADNP, HOMEZ PRD class, OXCART, LGFX, ARX, DUNX, DPRX, DRGX, DUX, DUXB, DUX 1, 2, 3, 4, 5, ISX, GSC, SM, HOPX, ISX, LUTX, NIXL, NOBOX, OTP, OX, CRX, PAX, FOXUB, PIX, IX, RAX, MOX, SEBOX, SHOX, SHOX, UNCX, NKL class, Box, BSX, INX, HHEX, NANOG, BNTX, X1, X4, X2, X8, X1, X2, X3, X5, X6, X1, X2, X3. If you enjoyed this video, don't forget to give it a thumbs up and subscribe to my channel.